Okay. Good morning, everybody. I'm live now. I'm live, Motion?
Yes, you are live. Yes,
Okay.
you are live.
So you're live. Okay. Good morning, everybody. Yeah, sorry for this little. Let me pass through over the last 30 minutes on the activity of PDCline Pharma. We are a biotech company based in Belgium developing a new class of conservine based on an presenting cell line. So in a nutshell, the company PDCline is a spin-off from the French Blood Bank. We are today a team of 25 people, including seasoned executives from biotech and pharma. And in terms of funding, we get close to 32 million euros so far. And we raised recently a round B of financing of close to 20 million euros, led by Korea Investment Partner. So the technology we are working on is based on the discovery of the scientific founder that were working in one of the research lab of the French Blood Bank in France. In fact, he has identified in the blood of leukemia patients a very rare leukemia, a leukemia affecting specific cells from blood, plasmatic cells. And from the cell from PBM patients, he developed a master cell bank, GMP approved for clinical use, that we are still using today as a so this cell line, and I will show you in more details how it works, is very easy to expand. It's an immortalized cell line. It's the origin of the cell, the fact that it's plasmacytoid dendritic cells and use very interesting biological properties. And you will see we are very highly potent antigen presenting cell. And furthermore, it's a very versatile platform. In fact, we just have to load the HLA present on the membrane of the cell by GM representative of tumor antigens, and we've addressed any virtual tumor. So we are a clinical stage company. First phase 1b academic feasibility study in melanoma is now completed, and now we are ongoing phase 1/2 non-small cell. So is present. So the PDC, the melanoma with antigens added on the result has been published, and now we are really focusing on PDC lung and PDC neo, where we're working on shared neoantigens, which is in preclinical stage. So we signed last year, early 2019, a first licensing deal with LG Chem to develop PDC lung cancer vaccine in Asia. So LG Chem is well known. LG is known for, at least in Europe, for electronics and flat screen, etc. But LG Chem is the 10th chemical company in the world, and they have a growing life science divisions. And we signed a deal with them, exclusive license in South Korea, and exclusive option for PDC lung, the lung cancer. And the total deal value $100 million, plus significant tied royalties and net sales in it's only for Asia. So one of the assets of the company is definitely the management team. You see here the teams. So for myself, I'm a president and CEO. I've been a co-founder of several biotech companies in the past, especially in cell therapy. I have been involved in several M&A IPOs. Recently hired medical directors that were in fact for all the major immunotherapeutic registration trials. And we have in the CMC Claude Dedry, which is in charge of manufacturing and QC for our products. The scientific founder Joël Pluma is joined full-time the company. and there is as well Laurent Lévy that joined the company, which is the CFO. So you see here as well the board Jean-Paul Prils, who is the former global R&D director and senior executive of GSK Vaccine, so has been involved in vaccine almost all his career. And we recently appointed Monder Majoubi as chairman. That was in fact the senior executive of AstraZeneca Oncology, former Genentech and others. He's today the CEO of Inet Pharma. So the company made significant achievement over the last 18 months. So we get the accredited quality control and release activity of vaccine from the Belgium authority in January 2019. In March, we signed a deal with LG Chem for our vaccine PDCline. We get the authorization to launch the phase 1/2 clinical trial in France and Belgium. We hired the medical director, Shanna Debreuil, in August 2019. In December, we closed a 20 million B round of financing led by, as I said earlier, the multi-billion Asian VC Korea investment partner. We administered first patients with the cancer vaccine in February, strengthened the board of directors with an industry leader in the field as Monder Majoubi, and lastly, we met a recent press release where we announced the dosing of the first patient with high dose in cancer with the cancer vaccine candidate PDCline 01. So it has been a busy years, and several achievements were realized. Okay, so let's pass now to the technology. So how it works. As I said, this cell line, the PDC and the plasmacytoid dendritic cells, have been developed and put in a master cell bank where the cells are in liquid nitrogens and cryopreserved in bags. And from bags of this master cell bank, we can develop working cell bank to expand the quantity of cells. And from a bag of working cell bank, we just have to seed single-use bioreactor. The cells grow in suspensions in a synthetic medium. It's the Lonza serum-free medium that we are using. And after 9 to 10 days of culture, we produce billion and billion of cells. At the harvest, we split the cells according to the number of peptide we want to load on the cells. We load individually the cells on the peptide on the cells. Then we pool the cells, irradiate the cells to stop the proliferations. Then the vaccine is put in a specific container from aseptic technology. When the patient is ready, because it's resistant to cryopreservations, we just have to pass through a syringe through the cap to inject the cells to the patients. And then the dendritic cells go to the lymph nodes, what you see here, to prime naive T cells that become memory T cells, then expand, migrate to the tumor microenvironment to make the job, which is to kill the tumor. Okay, so we did different benchmark study to demonstrate the capacity of our cell line to expand CD8 cytotoxic T cells specific of antigens. So here you see one of example where we have selected as an antigens the melanase. So we load the PDCline with melanase, and then we compare with other dendritic cells coming from differentiated from myeloid cell from the blood of donor that are the myeloid DC, and we have autologous and allogenic DC that are loaded as well with melanase, and we compare by putting with PBMC of donor the capacity of the cell line and the benchmark to expand CD8 T cell specific of melanase by fax dextramer. It's done in vitro after 21 days of culture. And as you can see here, we are 20 to 200-fold more potent than the conventional approach, showing the potency of our cell line. We did as well study with neoepitope, the neoantigens, which is today a very hot topic. We have for that selected two mutated peptide in the literature. What you see in red on the left, it's where the amino acid are mutated, and due to the tumor, the mutations in the tumor, and these different tumors found for one of them in melanoma patient and over one lung patients. And we loaded with this neoepitope the cell line with each of these neoepitope, and we put in culture with the CD8 T cells of 3 healthy donors. And as you can see here, it's HT03, HT02, and 01. We can definitely expand the CD8 T cell specific of the neoepitope, and we have more than 150-fold increase for each of them, what you see on the extreme right. So showing that the cell line is really potent to expand shared antigen, but as well neoepitope. And knowing that in the blood of healthy donor, the quantity of this kind of precursor specific of neoepitope is extremely low. It's 1 out of 1 million. So showing the potency, once again, of our cell line. So today, the cell line is loaded through the HLA-A2 of the cells, so it's the majority HLA. There's minority HLA present as well, like the B7 or B44, but we have genetically modified the cell line to express additional HLA. For example, we have added B35 or A24, and we have seen that the cells is very effective to present A24 specific epitope or B35, and that is interesting because we can definitely enlarge the patient population we can treat with the cells. So today we load the cell line with GMP peptide representative of the tumor antigens because it's the easiest way, the more convenience, I would say, but we did as well genetic modifications to make endogenous expression by the cell line full antigens or polyepitope. It works quite well. We did as well LA operations through with tumor mRNA transfections, and here again it works quite well. So we really have a very versatile technology, and we can definitely maximize the potentials with our technology. So in terms of IP, we have 3 key pillars that protect the IP of the company. First of it, we have the master cell bank, which is unique worldwide. We have exclusive license from the French Blood Bank. It is once again master cell bank clinical grade, coming into the blood from one patient, so one clone of one patient we are using all the time. We have the second pillar is the patent. We have 3, 2 granted, protect all these applications, plus the new one that has been filed, which is PCT process. And the last pillar is all the know-how and data related to process, QC in vitro, the immunomonitoring assay, and all the preclinical data. So all these 3 pillars represent all the key protections we can have in terms of IP. So we did first clinical trials on melanoma patients. As I said, it was an academic clinical trial where we have treated 9 patients with advanced melanoma. So after second or third line of therapy, the advanced form, once again, of metastatic melanoma. And we vaccinated 3 times the patients with the vaccine loaded with 4 different antigens, peptide of antigens like tyrosinase, GP100 melanase, and MAGE3. And and we have tested 3 different doses, in fact, of the vaccine, 3 patients per dose. And the result was definitely promising. We have seen acceptable tolerability and safety profile. That was the primary objective of these trials. We didn't observe any allogenic rejections detected in any of the 9 patients infused 3 times. And last but once again not least, we have been able to see significant antigen-specific CD8 T cell response that were directly detectable in the blood of patients, specific of the antigens infused in the vaccine in the blood. And that despite we infused just 3 times, and we didn't co-administrate any checkpoint inhibitor with our vaccine. And we know that there is synergetic effect with the checkpoint inhibitor. For that, we have done experiments to demonstrate the synergetic effect, and we have taken the blood of melanoma patients. Melanoma tumor is a very hot tumor with a lot of mutations, and they're very high tumor-induced immunosuppressions. We have put in contact the PBMC of the melanoma patients with the cell line loaded with melanase. And as you can see here, after 1 week of culture, we have 15-fold increase in terms of CD8 T cell representative of the melanase. With the checkpoint inhibitor alone, we have almost nothing in terms of expansions. But when we combine both, the checkpoint plus the vaccine, it's where you see the nice expansions and synergetic effect with a 35-fold increase and a 2-fold increase synergetic effect due to the checkpoint. Okay, let's pass now to the lung cancer trial that we are currently running. So, in fact, this is here the 6 antigens we have selected that are loaded on the vaccine for the lung cancer. It's non-small cell lung cancer. And so it's MAGE-A4, MAGE-A3, MUC1, NY, ESO1, survivin, and multimage. And what we did first is to demonstrate that for each of these antigens, a specific CD8 T cell expansion for each of these antigens with the cell line loaded with the antigens. And you see on the bottom, on the up part, the precursor before by fax dextramer. In red, it's what is specific CD8 T cell specific to each of these antigens. And at the bottom, it's after 2 weeks of culture with the cell line loaded with the antigens. And as you can see, we have definitely a very nice expansion for all of these antigens. For some of them, like the MAGE-A3 or the survivin, the expansion is a lot stronger, which is something we observe in immunology. But we have been able to expand all of them at a significant level. Okay, so what you see in this slide is the design of the current clinical trials. So we are going to, the main objective is to assess once again safety, because it's different antigens we are loading now compared with the melanoma trial, the immunological activity and the clinical response in association with checkpoint inhibitor. So we are going to enroll 62 patients in the current clinical trials. We'll have 4 cohorts of patients. We'll test, in fact, 2 doses of vaccine, 2 million cells per peptide for the low dose and 20 million cells per peptide for the high dose. And we'll start first with the vaccine alone in a adjuvant setting. And we will move for the 2 second cohorts with in first line metastatic in association with checkpoints. So the low dose vaccine alone monotherapy is already completed. And as I said earlier, we are now in a high dose monotherapy and as well in parallel in the in the low dose in association with checkpoint inhibitor. We will infuse 6 times weekly and through IV and subcutaneously infusions. Okay, just to finish my presentations, you will see the timeline of the activity. So we expect, we have started the clinical trial this year, and we expect to get the final result Q1 2023, according, of course, to the enrollment that has been a bit affected by the COVID situation. So the current clinical trial is ongoing in France and Belgium, and we expect to open Germany, centers in Germany soon. So we will get the clinical result in 2023 beginning and an interim report in Q3 2022. And we will carry on the work on the maximizing the value of the platform. So carry on the work on mechanisms of actions because we know why our cell line is so potent and why it's not. to optimize the PDC line, to work on neoantigens. So we are in the process to establish partnerships with some companies having expertise and engine for discovery of shared neoantigens. And we have as well going to work on the CMC development and optimizations. Today, we are working on bioreactor of 1 liters and 10 liters, and we would like to expand to the 50 liters bioreactor. So in 2023, we will have to prepare the second clinical trial that should be a phase 2/3 randomized clinical trials in association with checkpoints, and to set up that will be set up in US and in Asia at the top of Europe, and to work as well in over indications, new phase 1/2 in over indications. And that's what I wanted to present you today for my presentations. And you see here a nice picture of part of the team based in Belgium and France. I would like to thank you for your attentions. I don't know if we have time for questions now, if any. Hello?
Hello. Thank you so much, Eric, for your wonderful, insightful presentation. Thank you so much for your time, and thank you for being such a wonderful, phenomenal keynote speaker in our content. Uh, I would like audience to have to ask questions if they want to ask about your presentation. Uh, Eric, can you hear me? Hello.
Yes,
Yeah,
I can
perfect.
hear you.
So now finally, I will ask people to ask questions from Eric, and Eric kindly respond to them. And now we will be moving towards next presentation. But before that, I will show a video of our partner Pivot Path. Thank you so much. Thank you.